In today's video, we are going to learn about synthesis of thyroid hormones. Synthesis of thyroid hormones takes place in thyroglobulin, present in the follicular cavity. Iodine and tyrosine are essential for the formation of thyroid hormone. Iodine is consumed through diet and it is converted into iodide and absorbed from the GI tract. Tyrosine is also consumed through diet and is absorbed from the GI tract. For the synthesis of normal quantities of thyroid hormone, approximately 1 milligram of iodine is required per week or about 50 milligram per year. Since iodine is required in minute amount to prevent iodine deficiency, common table salt is iodized with 1 part of sodium iodide to every 1 lakh parts of sodium chloride. Synthesis of thyroid hormone occurs in 6 stages. Number 1, thyroglobulin synthesis. Number 2, iodide trapping. Number 3, oxidation of iodide. Number 4, transport of iodine into follicular cavity. Number 5, iodination of tyrosine. And number 6, coupling reactions. Now, the first stage of synthesis is thyroglobulin synthesis. Endoplasmic reticulum and Golgi apparatus in the follicular cells of thyroid gland synthesize and secrete thyroglobulin continuously. Thyroglobulin molecule is a large glycoprotein containing 140 molecules of amino acid tyrosine. After synthesis, thyroglobulin is stored in the follicle. The second stage is iodide trapping. Iodide is actively transported from blood into follicular cell against electrochemical gradient. This process is called iodide trapping. Iodide is transported into the follicular cell along with sodium by sodium iodide symport pump. Sodium iodide symport pump is also known as iodide pump. Normally, iodide is 30 times more concentrated in the thyroid gland than in the blood. However, during hyperactivity of the thyroid gland, the concentration of iodide increases 200 times more. Third stage is oxidation of iodide. Iodide must be oxidized to elementary iodine because only iodine is capable of combining with tyrosine to form thyroid hormones. The oxidation of iodide into iodine occurs inside the follicular cells in the presence of thyroid peroxidase. Please note that thyroid peroxidase enzyme is essential for the proper functioning of thyroid gland. Why? Because absence or inactivity of this enzyme stops the synthesis of thyroid hormones. Now the fourth stage is transport of iodine into follicular cavity. From the follicular cells, iodine is transported into follicular cavity by an iodide chloride pump, which is also called as pandrin. Now the fifth stage is iodination of tyrosine. Combination of iodine with tyrosine is known as iodination. It takes place in thyroglobulin. First, iodine is transported from the follicular cells into the follicular cavity, where it binds with thyroglobulin. This process is known as organification of thyroglobulin. Then iodine combines with tyrosine, which is already present in thyroglobulin. Iodination process is accelerated by the enzyme iodinase, which is secreted by follicular cells. Now, this iodination of tyrosine occurs in several stages. First, tyrosine is iodized into monoiodotyrosine, which is known as MIT, and later into diiodotyrosine, which is known as DIT. Now, MIT and DIT are called the iodotyrosine residues. Now, the last stage of synthesis is coupling reaction. Iodotyrosine residue get coupled with one another. The coupling occurs in different configurations to give rise to different thyroid hormones. One molecule of DIT and one molecule of MIT combines to form triiodothyronine or T3. Sometimes one molecule of MIT and one molecule of DIT combines to produce another form of T3, which is called reverse T3. two molecules of DIT combine to form tetraiodothyronine or T4, which is also known as thyroxine. So, this was all about the synthesis of thyroid hormones. If you like the information, then please subscribe to my channel, and in the comment section you can also suggest me the topics you want the videos on. Thank you.